don't do that. Hang on. Okay. Oh, let's start with a quote. Got to always start with a quote. As long as the brain is a mystery, the universe, the reflection of the structure of the brain, will also be a mystery. So, and the brain will probably always be a mystery to us. But this is by Santiago Ramon y Cajal, who's a very famous neurophysiologist who looked a lot of the structure of the nervous system and a lot of early neurophysiology experiments. So, very famous neuroscientist. So we're studying neuroscience. What is it? It's relatively new science. It's concerned with the development of the nervous system, the chemistry, the structure, the function, the pathology. It kind of puts it all into one, okay? As opposed to looking at pure physiology or pure anatomy, it's putting it together and seeing how the nervous system works as a whole and how it influences things like movement and thinking and all behaviors. There's lots of different areas in neuroscience, though. There's the molecular side of things, there's the cellular, the systems, behavioral, cognitive. We're going to call ours the clinical or physical medicine and rehab clinical neuroscience, get real specific here. So we're focusing heavily on motor and sensory systems. I would love to go down that rabbit hole of cognition and memory and all that stuff, but it fascinates me, but it's just too big of a topic to go into. So the organization and plan for this course, we're going to start with a real basic cellular overview, what happens at the cellular level with the nervous system. Then we'll talk a little bit about the development, in utero, and how that happens, because it has some implications for the adult nervous system, obviously, and helps you understand why things are in certain places when you look at anatomy and cross sections. If you know that developmental process, it'll help you with that. Then we'll look at what we call functional systems of the nervous system, like we'll do mainly sensory, motor systems, autonomic nervous system. Those are the three main functional systems we'll look at. Then we'll do a regional overview. So after we've looked at the functional systems, which actually go throughout your nervous system, like your motor system is in your cerebrum, your brainstem, and your spinal cord, but then we're going to step back and we're going to look at cerebrum by itself, brainstem by itself, spinal cord by itself, which has these functional systems running through it, but we're going to do a little deeper dive on the regional systems. We'll look at little special functions. We'll do like vision and vestibular systems are the main two that we'll do as far as special functions, special systems that are more relevant to PT. And then we'll look at support systems, which is going to be like your blood supply and also your coverings of your nervous system, your meninges and those kind of things. So those are kind of and that's kind of how you'll see this is broken up into those categories. And then that's kind of our stopping point for different exams is after each of these areas. Fun facts, human brain weighs about 3 pounds. It's about 2% of our body weight, but it receives 30% of our blood supply. So there's a lot of activity, metabolism going on in there most of the time. The brain itself, once you take off somebody's skull, they can wake you up from surgery and start poking in your brain, and they do those surgeries to make sure they're not, causing dysfunction when they're trying to do something for epilepsy or do some deep brain stimulation. You don't have any pain in your brain itself. You have a lot of pain sensors in your meninges, the covering of your brain, also the bone and skin. But once they get through that, they can kind of wake you up, and you won't feel much pain while they're doing those surgery. This is just kind of hard to wrap your head around here, but there's 100 billion neurons in the brain, and if you actually lay the neurons, you take them all out of your brain, you lay them side by side, not end to end, just side by side, like the axons and dendrites, right next to each other, and lay them. It would go 600 miles long if you pulled them all out individually and laid them. I don't know what it would be end to end. A lot further than that. Over 1 quadrillion synapses or connections between your neurons. So that's 500 million connections per cubic millimeter. This shows you the processing and integration power going on there, and why we'll never figure this brain thing out totally. It's just immense, and it changes. That's the cool thing about PT, and that's the cool thing about a lot of these new imaging things is now we can see that we actually change the brain. We're treating the brain. We're actually. So maybe we should go back to the, doing the anatomy on real, because we're actually, changing, sizes of people's cortices and strengthening the connections in these certain pathways by what we practice we do with our patients. However, here's another thing. Most of the cells in your brains are not neurons. Okay, you have 100 billion neurons, but there's a whole bunch of other cells that aren't neurons. These glial cells, the glial is glue, macrophages and astrocytes and those kind of things that are the support cells for the nervous system. So I don't know what brain freeze. Oh, I don't know. So I guess I was maybe. This is a fun fact about when you're drinking a Slurpee or something like that, or whatever you want to call it, a tasty freeze, and you get that headache, that brain freeze. It's called. If you want to get technical about it, it's sphenopalatine ganglioneuralgia. So you got to remember that, because the next time your friend has a brain freeze, you just got to break that out and say, oh man, that must suck. You got sphenopalatine ganglioneuralgia. What? Just totally freak them out. So that's a good word. And the other word I'll tell you is dysdiadochokinesia. That's just inability to rapid alternating movement. So you got some dysdiadochokinesia going on there when you see your friend at the bar who can't, do this, at a certain point. Do we use only 10% of our brain? There's have you ever heard that, you only use 10% of your brain? No, you use all your brain. Most of the time, you may, obviously when you're concentrating on things, you may use a certain region, but your brain is active. I wish it was that way. That would give me some hope that I could improve it, but I think I'm already running at full capacity right now, so. All right, let's take a break here. Pause this. Let's just start with, I'm a big picture person. I want to see the big picture and how parts relate to each other before I start to dig in. So, come on. And I knew I was going to forget one thing to print out. I normally print this out for you guys, and I will print a copy out to give you tomorrow. Oh, sweet. Thank you. It's at the last page of your outline. See, I'm losing it, man. I don't even remember stuff I gave you. So, thanks. So there is a little thing there. This just helps me understand how different parts of the nervous system are related to each other. So you just work yourself left to right on this. You got the nervous system. We can roughly divide it into the little neurons and the glial cells, the actual cellular components of it. And we're going to have these major functional systems or components called the autonomic nervous system, the peripheral nervous system, and the central nervous system. Now the autonomic nervous system is both in your central and peripheral nervous system, but we're just going to pull it out there make it just for categorization. It makes it a little easier to look at this. So then if you keep going to the right, the autonomic nervous system basically breaks down to a sympathetic and parasympathetic. The peripheral nervous system is just your cranial nerves and your spinal nerves. Then the CNS, you move left or right there, and you got your brain and spinal cord. And then the brain breaks down to the cerebrum, the cerebellum, and the brainstem. The brainstem is actually part of your brain, okay, when you talk about the brain. So those are the three components of the brain. And then you go, I'm not going to follow this whole thing out, but this gives you the picture that's just part of that part. That's a subcomponent of that, okay? So just important to kind of understand that big organizational structure just logically, and when you're trying to guess a question on an exam, some of this big understanding will help you probably that's the answer because I know that's kind of a component of that or that kind of thing. So got it? So we're going to go through some of that right now. So this is the major components of the nervous system is the neurons and neuroglial cells, the central nervous system, peripheral nervous system, and autonomic nervous system. So the neurons, the basic functional structural unit of the nervous system. These are the things that convey information into the central nervous system and also help with processing within the central nervous system when they communicate with each other. If you have a neuron that brings information into the nervous system, okay, or a neuron that's bringing it into a structure within the nervous system, it's an afferent neuron. I think of the A in there as acquiring information. I'm acquiring. I'm afferent. I'm acquiring. Information is coming to me. I'm an afferent neuron. It's traveling towards a structure, to do something. If a neuron is going away from that structure, going to another structure, or to a muscle, or to a gland, or something like that, to tell something what to do, or to affect another structure, if the neuron is sending an impulse, an action potential away from it to go to another place, that's an efferent neuron. And I think of efferent as exiting. It's leaving that structure. It's efferently. It's going away. So an efferent neuron. Neurons that connect only with other neurons are called interneurons. And actually, interneurons are the majority of all your neurons. We're going to spend a whole lot of time focusing on the major sensory and motor neurons, which go to, your motor neurons go to connect with muscles. That's your major efferent neurons. And then your sensory neurons come from peripheral sensors, thermoreceptors and mechanoreceptors, and bring it to your central nervous system on these long axons, relatively long axons. Those are what we typically are calling efferent neurons, or like your vision, those neurons that come from your eye or from your inner ear. Those are afferent neurons bringing information towards the central nervous system to be processed. But we're going to spend an inordinate amount of time talking about those, but the interneurons, which are the majority of your neurons, actually take all that information, process it, make sense of it, decide how to respond to that. The other big classification of cells within the nervous system are what are collectively referred to as the neuroglial cells. Okay, and actually, remember we said you have like 100 billion neurons, but you actually have a lot more glial cells. These are supporting neurons that they don't necessarily convey information. They don't generate action potentials. They don't have the ability to rapidly change charge and reestablish that charge after it's been changed. They just maintain a certain level of base charge. Now, they may regulate up and down a little bit. There's actually new research showing that these glial cells may play a bigger role in information processing than we thought. But we're going to, like right now, just say that they're kind of support neurons. They provide support services. They help to insulate the nervous system. They help to nourish, provide protection, help to manage waste, get rid of old stuff, deal with bacteria and viruses and those kind of things. So those are the glial cells. So now let's talk about this. Let's focus on just the central nervous system briefly here. Generally, there's considered to be 7 main parts to the central nervous system. Okay, these would include your cerebral hemispheres, the things you typically think of when you think of the brain, but that's the cerebrum. It's the cerebral hemispheres, these big guys up here. And inside of those is some big, we call it nuclei. A group of cell bodies within that is deep inside of nervous system tissue are generally referred to as a nuclei if it's in your central nervous system. So the basal ganglia, also referred to as the basal nuclei. Ganglia is, we'll get to that in a second, but they call it the basal ganglia, but some people are trying to change it to basal nuclei because it's really a group of cell bodies within the central nervous system are typically referred to as a nuclei. This is an exception. So basal ganglia are just a group of cell bodies, and we'll get to the basal. You'll know more about the basal ganglia than you ever wanted to in a few weeks. The diencephalon, which is the thalamus and hypothalamus together, di is 2, diencephalon. Encephalon within the cephalic structures, it's right in the middle of your brain. There is the thalamus and hypothalamus. That's considered one part of the central nervous system. This thing collectively called the diencephalon. Then we have the brainstem, which is the midbrain, pons, and medulla. We also have the cerebellum sticking off the back of that, and then finally the spinal cord. So those are generally what textbooks consider as like the main parts of the central nervous system: is the cerebral hemispheres that include the basal ganglia, the diencephalon, thalamus and hypothalamus, also stuck right in the center, the midbrain, the pons, medulla, cerebellum sitting off the back, and the spinal cord. So that is these guys right here, okay? I wonder if I remembered to. So my little drawing thing here. So cerebral hemispheres right here. Basal ganglia you cannot see. They're deep within the cerebral hemispheres, okay? You can't see the basal ganglia in this view, okay? But they're deep within there, okay? The diencephalon, which is, so this is a sagittal section right through my brain. Somebody's cut right through here, mid-sagittal. There's that little split between your two cerebral hemispheres. That's what we've cut right through, and we're looking at the inner surface of one side of my brain. So this is a right hemisphere looking at that, surface of my right hemisphere, that inner surface. So that's the diencephalon, which is the thalamus, is the, like right here, the egg-like, and then this is hypothalamus. So hypo below the thalamus, and this is a little bit anterior, and below the thalamus. Okay. But that whole structure together is a diencephalon. Okay? Then we get into the brainstem. And here's the midbrain. And if you look at it, that's a good name, midbrain, because if you consider the brain, all this structure here that includes the cerebellum and the brainstem, it's kind of in the middle. That's the midbrain is right there. Then the next structure in the brainstem is this thing called the pons, and it's got a little bit of a bulbous, rounded front part to it. That's called the basilar part of the pons. I'm going to just say things that you're going to hear again and again. Doesn't mean you'll have to write everything down, but just starting to get you to hear things repetitively. I'll mention things I don't necessarily expect you to know all right now, but just going to, but I'll try to tell you what you need to know this. So this is just kind of getting you familiar with terminology. Then the medulla right here below that. And then finally the spinal cord down here. That changes names. Here's the foramen magnum. That's the opening in the bottom of your skull, the foramen magnum. That's how you know you're on the spinal cord now. There's not like an actual line there with any of these things. It's just, you got to know based on different landmarks. And then you got your cerebellum sitting back here. Those are the 7 main parts of the nervous system right there. That's the central nervous system. Now peripheral nervous system. This is what you guys have studied. You know the peripheral nervous system better than I do right now. Like you guys know your nerve roots going to those muscles probably, and you know your brachial plexuses and your lumbar sacral plexuses better than I do right now. So that makes you feel better. Good. Enjoy it while it lasts, because we'll be getting into stuff you won't know better than I do it soon. But I'll give you that one. So the peripheral nervous system is all parts of the nervous system not encased in the skull or vertebral column. So basically your cranial nerves and your spinal nerves. So I tell people, like, people get all freaked out sometimes on the cranial nerves. They're just like your spinal nerves. They're just going to your head, face, neck, and also they subserve some special functions, vision, vestibular, olfaction, that kind of things. They have a few special functions, but they're really like peripheral nerves, just like the ones coming out of your spinal cord. These are just coming out of your brainstem. Okay? So the cranial nerves and the spinal nerves. So on that picture, everything in yellow is your peripheral nervous system, and everything in blue, or whatever that color is, chrome, blue, something like blue, is your central nervous system. Okay, and your peripheral nervous system is just is mainly a bunch of axons, long axons, and most of your peripheral nerves, except for some of the cranial nerves, contain motor, sensory, and autonomic, axons in those that are going to and from your peripheral to your central, structures and so forth. So that's what you need to know about peripheral nervous system. So every one of those big nerves, you cut through it, there's lots of little axons in there, and they are typically a mix of motor, sensory, autonomic. Except for some of the cranial nerves, that's where it gets a little more specific. Okay? And the autonomic nervous system I mentioned is contained both within your central nervous system and peripheral nervous system. So there's the control centers for your autonomic nervous system are mainly in your hypothalamus. Of course, your pituitary gland plays a role in that as well. And then there's pathways running down through your brainstem and spinal cord that goes to talk to the peripheral nerves, your autonomic peripheral nerves, cranial and sacral peripheral nerves, and thoracic lumbar division here, the sympathetic and parasympathetic nerves tell them what to do. Okay, so typically we have a sympathetic and a parasympathetic division. So the sympathetic is that we call fight or flight system, tends to ramp up things, make you more vigilant, increase heart rate, increase respiratory rate. It's also referred to as a thoracolumbar division because the nerves that go to do that type of sympathetic responses tend to exit out of your spinal column in the thoracolumbar region. Okay, and then they go from there. If they need to go up to your head or neck, they can exit the thoracolumbar and then travel up or travel down. Okay, to do that. The parasympathetic division, the need and feed system, is also called the craniosacral division because the nerves for your parasympathetic system exit from the cranial nerves and the sacral nerves. Okay, so you have cranial nerves that have some parasympathetic control. You have your big, huge vagus nerve that goes down, and it actually does almost a lot of your parasympathetic control of your organs and so forth through vagus nerve stimulation. And then you have your sacral division that does a lot of the parasympathetic regulation for your bowel, bladder, genitals, and those kind of things. That's why it's called the craniosacral division of the autonomic nervous system. And what are these? Generally, they innervate smooth muscle, cardiac muscle, and secretory glands. And their role is homeostasis, ramping up your system in the background, up or down as needed to maintain appropriate homeostasis, depending on what you need your body to do. And we'll go into much more detail. We'll have a whole session on autonomic nervous system. This is just a very brief schematic. Don't worry about this right now. We'll do this later. But just this is the parasympathetic, which here are the nerves that go to interact with those organs. For sympathetic control is exiting in through the thoracolumbar region. That's where those nerves exit. And then parasympathetic division here on this side, cranial nerves and sacral nerves that go to supply most of those same organ systems. They kind of counterbalance each other and oppose each other's actions in most cases, not all cases, but they tend to counterbalance each other. Okay. All right, let's talk about some just kind of just like anatomy is a language. I mean, neuroanatomy is a whole language in and of itself. And if you understand some of the basics language concepts of it and the roots of this stuff, you can figure out some of this terminology on your own or what the meaning is. And I'll try to. Then it really can help you with your reading and also with test questions and those kind of things by breaking down some of these words. And that doesn't happen all the time. And there's actually there's a book about this thick that I have in my office. It's called Alternative Neuroanatomical Terms. And so for every neuro term, there's like 6 alternatives. And so it's a whole textbook on just all the alternative terms. I try to make my terminology consistent with these 2 books, but there's even some discrepancy between these 2 books and terminology. This is out of the UK. So this is out of England, and there's a little bit difference sometimes in what they call things, but not bad, and so forth. So, but we'll try to come up with the most common. We'll try to establish our language that we're going to use here. Okay. So first of all, we need to talk about terms describe locations in the nervous system, where some part is relative to another part, just like you did with an anatomy. You got to say if something's distal or proximal or dorsal or rostral or those kind of things. So the nervous system is very similar, okay? But there's a little bit of a caveat to the nervous system, which we'll get to in a second here. So these are the terms. Something superior, the part is typically situated above another part, superior. Inferior, the part is below another part. You guys heard the term rostral before? Okay, rostral is toward the head or the beak, okay? So rostral is more towards the beak or the face or the head of the person. That's in relationship to the neuraxis. So when we talk about the terms rostral and caudal, we're referring to the neuroaxis or neuraxis, not the longitudinal axis through the body, which we'll show you a picture here. It'll make sense in a second what I mean by that. So rostral is towards the head or beak as it relates to the neuraxis. Caudal is towards the tail or coccyx as it relates to the neuraxis. If you use the term anterior, that's towards the front of the body. That's referring to the long axis of the body. You know what anterior is. Ventral is towards the front, but that's relationship to the neuraxis. Posterior is towards the back as it relates to the axis of the body. Dorsal is towards the back as it relates to the neuraxis. So And ipsilateral means that something begins, like a tract or a pathway, begins and ends on the same side. So as long as that pathway begins and ends on the same side, it's an ipsilateral tract. And there are a few tracts that start on one side, cross over, then cross back over. But if they start and end on the same side, it's still an ipsilateral tract. If a tract starts on one side and ends on the other side and stays there, that's a contralateral tract. Okay? So in the neuro world tends to use the terms rostral, caudal, ventral, dorsal when they're talking about how to refer to where structures are within the nervous system as they relate to each other, because those are terms that are related to what's the neuraxis versus the body axis. But a lot of books will also use anterior, posterior, superior, inferior, and I may use those too, but you have to just know where you're at in the nervous system, and then it all makes sense. So let me show you pictures here. I like pictures. So if you're a four-legged animal, like a crocodile here, and your axis through your long axis through your body and your nervous system, so this crocodile's brain, spinal cord, and everything is lined up all along its long axis of its body, okay? And so that in that case, the terms always are synonymous to each other and match up. Like rostral is the same as anterior, ventral would be the same as inferior. Although it doesn't show that there. Dorsal would be superior, caudal would be posterior. But ignore the crocodile now. I'm just showing you that's what is the neuraxis in four-legged animals, where they're down like this, typically is in line with their body axis. Everything's lined up because their nervous system's in that. Humans, however, and this is when we talk about development of the nervous system, you'll see what happens here is our brain and spinal cord all start out in this just long tube that are all lined up. As we start to develop, because we're two-legged animals, eventually what happens is as our brain starts to develop, it kinks over. It like tips like this, because our eyeballs are going to be out here and we're going to be on two legs and we're looking forward. And so this is called the cervical flexure. So the brain and brainstem kind of tip. It's really the brain, it's the cerebrum basically, just kind of tips over and gets a little kink in it. It's not a 90 degree. It shows it a 90 degree here. So the neuraxis, till you get to the cerebrum, so the neuraxis through the spinal cord and the brainstem matches the body's longitudinal axis. Okay, so if I say something is posterior, in the spinal cord, that's the same as dorsal in the spinal cord. To the dorsal, like the dorsal fin back here, dorsum. Okay, but if I get up and if I know I'm in the cerebrum, and I say something is dorsal, it's actually superior in the cerebrum. So I could say a structure is superior or dorsal, and they would mean the same thing in the cerebrum. Down in the spinal cord, if I say if something is dorsal to another structure or posterior, then those match up. So the first question, if I'm asking you a question about where a structure is located, you have to ask yourself, all right, where am I at? Am I in a spinal cord or brainstem? Then everything matches the body, like those terms are synonymous. But when I get up into the cerebrum, everything tips like this. So this is a better picture here. So let me give you an example here. So let's say, in the spinal cord, you have these things that come off. Your spinal nerves are formed by a dorsal and a ventral root, right? There's two roots that come together to form the one nerve. And so you have a ventral root coming off of your spinal cord. But you could also call it an anterior root, because ventral and anterior are synonymous there, because those are in line with the axis of your body. This is anterior, and that's the same as ventral, okay? However, let's go up into the cerebrum, and the cerebellum follows the same rule too. So cerebellum and cerebrum, this is that cervical flexure. If I say the hippocampus, which is down in the temporal lobe here, the hippocampus, I'll draw it on here. Hippocampus is right in there. And then here's your basal ganglia is here. If I say in the cerebrum, I would say the hippocampus is ventral to the caudate or whatever, some part of the basal ganglia. What other term could I say then for in the what also would the hippocampus be? It's ventral or inferior. So here ventral and inferior are synonymous, where here ventral and anterior were synonymous. Does that make sense? So you just got to like ask yourself, because I could ask a question about relationships between where structures are. Is one superior to another one? Is one inferior to another one? Is one dorsal? Is one ventral? Is one rostral or anterior? I should be able to use any of those terms, and as long as you know where you're at, as long as you know like, oh, those are terms that are referring to cerebral, those are cerebrum terms, then like, so here's, you know, rostral is towards the beak, towards the front here. However, if you extend this out like this, straight up here on a four-legged animal, rostral would be up here. But our neuraxis tilts here. And so in a human, you know, my frontal lobe is rostral to my parietal lobe, or it's anterior to my parietal lobe, okay? But down here in the brainstem, I would say, here's the pons right here. The pons is, I could also call that rostral. So the pons is rostral, because this is still in the body axis, and this will still be rostral. This hasn't tilted yet. The axis hasn't tilted. So the pons is rostral to the medulla, or it's superior to the medulla. We're up in the cerebrum, you know, this frontal lobe is rostral or anterior to the parietal lobe. It's basic concept. I mean, I'm probably making it harder than I should, but it is a concept you should understand, because as you're reading, it's really important when they're talking about like this structure is above this one. You can get totally lost and confused if you don't understand that basic concept when you're trying to see where, especially when we start talking pathways and when things are connecting to other things and all that kind of stuff. So come to my office if you need to talk it through, and yeah. Good question. So the pons actually, it's a structure like that. So there is a portion. So this is the basilar pons right here. That's little. So this is the medulla. This is a good point. So the basilar pons could be both rostral, superior, as well as a little bit ventral and anterior. So that basilar portion of the pons is a little anterior rostral, ventral, superior. You could use those terms, but that's getting a little more nuanced. So you guys are too smart, thinking too hard. Don't go that deep yet. Come on, first day. All right. Anatomical planes, you guys know this stuff. Frontal plane, which is the coronal plane. Sagittal, which is like, we're going to only talk about a median sagittal or mid-sagittal cut right through the brain, and then there's also parasagittal when you come off that median. But the only view you're going to see in our world is a median sagittal cut through the cerebrum and through the brain stem and the spinal cord. Now horizontal is this one, this cut horizontally. You'll see lots of those. We're going to see lots of horizontal cuts through the cerebrum, brain stem, and spinal cord. So those are your major planes of motion. The coronal plane is this one, this frontal plane cut down through this way, sagittal through the middle, horizontal this way, okay? So here's our classic this is a mid-horizontal section through the cerebrum, okay? Just cut right this way and looking straight down. It's a mid-horizontal. We're looking straight down through that cut. We've taken the top of the brain off here and looking down that way. Here's our classic mid-coronal. Cut straight down through this way, looking at the front of that. After we've taken the front of your brain off, we're looking in the middle of your brain there. And this is our mid-sagittal cut right here. That's the one that's cut straight down the middle between the two cerebral hemispheres. Okay, between the cerebral, it's called the cerebral longitudinal fissure. This big gap there. This would be a horizontal cut through the medulla, okay, through a section of the medulla. That's what you're going to see stuff, these little ink blotty looking things. You have no idea what they are, but you will. Believe me, you will. We're still on neuroterminology here. So let's talk about some general terms that are used to describe pathways or groups of nerve fibers, which are typically axons. So these are groups or bundles of axons. And so if you see these terms attached to anything, that means they're just talking about a bundle of nerve fibers, okay? So if you hear something referred to as a capsule, the one you're going to hear the most of is called the internal capsule. That's a big bunch of axons. This big white matter in the middle of your cerebrum, where a lot of your motor and sensory fibers are going to your cortex, is called the internal capsule. So if you hear a capsule, oh, that's just a group of axons. If you hear a radiation, that's also a group of axons. The one we'll talk about most commonly is the optic radiation. So when information goes into your eye and first goes into your brain, it gets back to your occipital lobe via the optic radiations, okay? And it's very obvious to see, and we'll show you a cross-section of a brain, you'll see the optic radiations that are taking that information back to the... If you hear of a lemniscus, which means ribbon-like, it's kind of like ribbon is like a flattened thing, it's kind of flat. It's a flattened out, ribbon-like group of axons. And we'll talk about one of your major sensory pathways is called the medial, it's called the dorsal column / medial lemniscal system, okay? So the medial lemniscus is these axons that are carrying proprioceptive information through your brainstem at that point, so the medial lemniscus. A tract, anytime you hear referred to as the, a tract, a spinal thalamic tract is a pain and temperature pathway in your spinal cord. That is just a bunch of axons, sensory axons, okay? If you hear the term a fasciculus, we're going to talk about things like the fasciculus gracilis and fasciculus cuneatus. Again, don't need to write these down, just giving you examples. Those are also sensory pathways that are carrying, those are part of your dorsal columns, these columns of pathways that are called the fasciculus, fasciculus gracilis and fasciculus cuneatus. A funiculus is larger than a fasciculus typically, and so we're going to talk about the funiculi or the funiculus of the spinal cord. The spinal cord has a bunch of pathways, like it has some of these smaller tracts and things running into it, but we can divide the spinal cord into some bigger packs called funiculi, like the posterolateral funiculus, the anterolateral funiculus. That's a bigger thing that some of these other smaller tracts are running in. So a nerve. Anytime you hear a nerve, that just means there's a bunch of axons in there, okay? Could be sensory, could be motor, could be autonomic, could be combination. Most of the time it's a combination of those. Unless it's like your optic nerve or your vestibular nerve. A commissure, if you hear a commissure fibril is where two things join. A place where two things join is a commissure, okay? We'll talk about the anterior white commissure. There's a posterior white commissure. There's a posterior commissure. Those are, like the anterior white commissure is actually these fibers that run from one temporal lobe all the way to the other temporal lobe on the other side. So left and right temporal lobes are connected through these commissural fibers called the anterior white commissure. But again, it's just axons, okay, groups of axons. So that was axons. And there's also some common terms that used to actually describe where the cell bodies are, where these neuronal cell bodies are located that have axons that either are coming into them or going out of these cell bodies, right? And so these are some of the things you'll see. A ganglia, okay? If you hear the term ganglia, that's typically referring to a group of cell bodies that are all close to each other. Most of the time, a ganglia is a group of cell bodies you find in the peripheral nervous system, with the exception of the basal ganglia, which I said that are trying to change the basal nuclei. So in the peripheral nervous system, the most common one you'll hear is dorsal root ganglia. That's where some sensory cell bodies are located. Your first order neurons that are bringing in sensory information from the periphery, they're actually located outside your spinal cord, called the dorsal root ganglia. Or you might hear of like terms called the cervical ganglia. Okay, there's some ganglia that are associated with some of the autonomic nervous system going up to into your face to supply that, where some of the cell bodies are located outside in the cervical ganglia. And so forth. So if you hear ganglia, that's typically referred to a group of cell bodies in the peripheral nervous system, with the exception of the basal ganglia or basal nuclei in the central nervous system. A lamina, if you hear the term lamina, that's a, do you know what laminated is? Like a laminated board is like multiple layers. There's places like there's within our spinal cord in the gray matter, the dark matter, where the cell bodies are, which are in the middle of our spinal cord. There's these called Rexid's lamina. There's different types of cell bodies of neurons that are associated with different types of function that gives it a laminated appearance under a microscope, because they're different, because they subserve different functions. The structure looks a little slightly different, so they're laminated in there. They call them Rexid's lamina. So we'll talk about these different lamina number 1 or lamina number 2 in the spinal cord. Okay, so don't have to remember that, just giving you examples. A body. You know, if you hear the term body, you know, nuclear body, cell body, something like that, that is also typically referred to as a group of nuclei. And then nucleus. If you hear the term nucleus, you're not talking about the nucleus of an individual cell if we're referring to a structure. If it's a nucleus, like that's why the basal nuclei is what they're trying to change that to. Or we have the cuneate nucleus, so we have the fasciculus nucleus, or the cuneate nucleus and the gracile nucleus, I'm sorry. Those are cell bodies that are located in the brainstem that we'll talk about. So, or the acoustic nucleus or the vestibular nucleus, those are just cell bodies. Getting there, more neuroterminology. Furrows and depressions. So we're talking about here typically things on the surface structures of your nervous system. And these are similar to some of your anatomy things. Well, so sulci. A sulci is a rut or a furrow. So all the spots when you look at your brain all wrinkled, that's the depression. So the depressions in your brain are the ruts, the furrows, those are the sulci, okay? A fissure is something that's even a little larger than a sulci. So the big one between your two cerebral hemispheres, that's called the cerebral longitudinal fissure, okay? Or along the side of your brain, there's a thing called the lateral fissure, which we'll look at. That's a little bigger than a sulci. Really, you'll see it tomorrow on a real brain, okay? A fossa. You guys have learned fossas in bones, so it's the same thing. It's just a defined depression, okay? So we'll talk about some fossas when we get to those. Bumps and ridges. So bumps, protrusions from your nervous system. A peduncle, okay? A stem-like connecting part. We'll talk about the cerebral peduncles. That's the connecting part from your midbrain up to your cerebrum. When you look at the front of your midbrain, those are called the cerebral peduncles, okay? A gyrus, okay? So you got the ruts and furrows, but when the other parts of their brain, the actual rounded parts that are bumping up there, those are the gyri or gyruses, okay? Which means circle. And a brachium, you guys have learned brachiums, right? But brachium is an arm, okay? We'll talk about the brachium of the inferior colliculus, which is a relay structure for auditory, when auditory information initially comes into your brainstem. One place it goes is the inferior colliculus, and then there's a brachium that takes it up to go up into your central nervous system, called the brachium of the inferior colliculus. It's just an arm. Tubercles. You'll hear about tubercles. We'll have a gracile tubercle. We'll have a cuneate tubercle. This is a bump or nodule or small eminence off. This is something on the surface of your central nervous system, or on. It doesn't have to be central nervous system. There's a bump or small tubercle or eminence. Layers. We have layers in our nervous system. So we've referred to laminas, which are groups of cell bodies that are laid down in this kind of laminar type fashion, which means thin sheet or veneer. The cortex. The cortex is the outside of our cerebrum, that little tiny layer. That's where all those cell bodies are. Most of the rest of your cerebrum is white matter, is axons. That's cortex, sorry. We'll refer to a thing called the tectum of the midbrain. It's like the roof of the midbrain, and I'll give you, it's hard to, you kind of have to imagine this roof, but it is kind of like a roof of the midbrain, the tectum of the midbrain. It's a layer of the midbrain. We can divide our midbrain into layers when you look at it sagittally, but don't worry about it. And also a tegmentum is called covering. There's a tegmentum of the brainstem that we can talk about as well. Those are layers. Hanging in there? All right, let's get started with some anatomy now that you know the terminology. Now we can start looking at structures and some basic anatomy. We are going to start with the cerebrum. Big overview here, big picture. We're going to have a whole session or 2 days, I think, on cerebrum later on, but when we get to regional areas. But this is just, these are the most important things to know. This is just really like, hey, know the basics first. Cerebral hemispheres, these big guys up here. Excluding, we're not talking brainstem here, we're just talking cerebral hemispheres. What do? They process things, right? They process sensory, motor, memory. They're also important for your basic reasoning, language, intelligence, personality, emotion. All that happens in the cerebrum, okay? And it's the thing we probably know the least about. You know, we know probably more about brainstem and spinal cord and all that, and least about cerebrum. It has 4 primary lobes that we talk about: a frontal, parietal, temporal, and occipital lobe. Probably most of you have heard those terms before. Some people talk about a 5th lobe called the limbic or insular lobe, which will show you where that is. So just in case you're reading and somebody talks about the limbic lobe, you're like, what the hell is a limbic lobe? There is actually a limbic or insular lobe. Again, those limbic and insular are interchangeable there in that particular terminology, but most people call it the limbic lobe. There's a lateral sylvian fissure, big, you know, called lateral fissure on the side of your brain between your temporal lobe and parietal and frontal lobes. Okay, we'll show you that. This will be on pictures in a second. There's a corpus callosum, which is that big connecting group of axons that connect your left and right hemispheres, okay, your cerebral hemispheres. There's this thing called the corpus callosum that connects those. So when you like take somebody's brain and you pull apart the cerebral longitudinal fissure, you see this really white shiny thing. That's the corpus callosum. And that's the thing they actually go in children and cut in half when they have really bad seizures that go, and that prevents the spread from one side of your brain to the other side of your brain. And you can do that, and the person can function pretty normally with the cut corpus callosum. You have to put somebody in very interesting, specific situations to find the deficit. So it's really kind of cool. We'll talk about that later when we get to the cerebrum. The surface has got the gray matter of your cerebral hemisphere. That's where all the cell bodies are, in the gray matter in the cortex. The deep to the cortex is mostly the white matter, except we do have a few groups of cell bodies located down, kind of stuck in that white matter, which is mainly our basal ganglia, okay, which consists of some subcomponents. The basal ganglia are a group of nuclei called the caudate, the globus pallidus, and the putamen. Also referred to as the basal ganglia could be called the basal ganglia, it could be called the cerebral nuclei, or it could be called the basal nuclei. And I'm sure there's probably 4 other ways we could talk about that, but most textbooks still call it the basal ganglia, and that's what we're going to refer to it as. But just in case you're reading some other resource and they call it the cerebral nuclei, you're like, what is the cerebral nuclei? It's the same thing as the basal ganglia. If they say basal nuclei, that's the same thing as the basal ganglia. Within the cerebrum, remember, there's also the diencephalon. Okay, that's one of your 7 main parts of your central nervous system. That's primarily what we're talking about. The diencephalon is these 2 guys, the thalamus and the hypothalamus. Okay, they do a lot. They're small, but they do a lot, and they're very centrally located. You know, that's important. Structures are centrally located because then they can communicate and affect lots of different areas. So the thalamus is really a major relay center for sensory and motor. Most things that go into your brain have to stop by the thalamus first. It's like the secretary of your brain. If you're bringing in information, like afferent information coming in, it's got to go to the thalamus first. And when other structures are talking to each other within your central nervous system, oftentimes they make a stop at the thalamus. When we talk about the basal ganglia and how it influences movement and so forth, one of the stops it has to make is it has to go talk to the thalamus. So it's a kind of a governing structure, relay structure. Okay, the hypothalamus, this is the thing that's controlling your autonomic nervous system, your homeostasis, reproduction, eating, and growth. The hypothalamus is important for regulating all that. It has direct connections with your autonomic nervous system. It's the major governor of your autonomic nervous system. Okay. don't have to know these now, but I'm just pointing them out. So here's another thing with an alamus at the end. So you've got all these things that have an alamus, alamus, alamus. There's an epithalamus, which is your pineal gland. Sits off the back of your thalamus. It regulates, has some regulation of the pituitary, influences the pituitary, it produces melatonin, for sleep regulation, and it also influences your adrenals. Okay, so it also regulates some autonomic function. It complements the hypothalamus, this epithalamus, or this pineal gland is what's often referred to, but it's epithalamus. Then the subthalamus is a subcomponent of the hypothalamus. However, it is actually involved. It actually becomes part of the circuitry of your basal ganglia as it regulates movement, even though it's a thalamus structure. So I'm just pointing these out so you don't get these confused. And just the diencephalon we're talking about is thalamus and hypothalamus. These other guys are not part of your diencephalon technically, but they're right around there. They hang out in the same territory in the same neighborhood. Just don't want you to get too confused with that. So here's your cerebral hemispheres. Okay. Deep inside the cerebral hemispheres, you have a pair of these. You have basal ganglia structure on both sides. That's what's in yellow. Everything in yellow is basal ganglia. This little orange guy is not part of the basal ganglia. Ignore the orange guy for right now. Okay? I'm not even going to name these parts yet. Just understand here is basal ganglia on the left hemisphere, and then there's a very small gap between. On the other side, there's the other basal ganglia in the right hemisphere. They're real close to each other. They're just like this, really close to each other, but you can't see them. You can't see them from the outside. You can't see them from the inside, or from the inside view. You got to cut down deep inside. They're stuck in the middle there. So they're kind of hanging out in the middle there. You can't see them externally, and you can't even see them with a mid-sagittal cut. You have to imagine them. That's why I'm showing you here these basal ganglia floating there in the middle of your white matter, in the middle of your cerebrum on both sides. When we get to the basal ganglia and we look at the different cuts, it'll start to make sense how these fit in there, and get that three-dimensional perspective. This little guy here, this little blue thing here, that's your thalamus. And you have a thalamus on each side. Most of your structures are paired in the nervous system on both sides. So you have your thalamus that are really close together right here, and then just sitting a little more lateral and a little more outside is those basal ganglia structures. The thalamuses are sitting right in the middle there. There's a ventricle system right here, right between the third ventricle. And when we cut right down through that and we pull your brain apart, we can actually see the thalamus on both sides. You have a left and right thalamus. We see it on both sides here, but the basal ganglia is a little bit deeper behind the thalamus. It's being hidden behind the thalamus. So you see that blue structure. There's another one right next to it on the other side. So you're looking at the outside of the left thalamus here, but there's a right thalamus right on the other side there, and it's separated by this little tiny gap between your two cerebral hemispheres filled with cerebral spinal fluid called the third ventricle, which is a little tiny sliver of a gap that's in there. So this is that mid-sagittal cut right here. We're looking at the inside surface. This would be a right hemisphere, the medial surface of the right hemisphere, cerebral hemisphere. And so you would have, here's your thalamus right here. That's the egg-like structure, okay? Then deep to that would be this kind of basal ganglia structure that's more lateral to the thalamus. That's kind of in that area, but you can't see it. It's back in there. Where's my little eraser here? This is the corpus callosum. And you'll see a picture on a real body here. Those are those commissural fibers. Those are commissural fibers that are connecting two halves, left and right, together. Corpus callosum. So here's the breakdown on looking at just the cerebral hemispheres, the lobular structure, the lobes of the cerebral hemispheres, starting with just the frontal lobe, everything in blue. parietal lobe, or actually temporal. Let's go temporal. Let's go in order what they have. Temporal lobe is right here in light orange there. Parietal lobe here in green. Occipital lobe back here. And when we look at when we get to the actual anatomy, and we'll do it on real, well, I'll give you how you define where those, what gives you to know where those begin and end, at least roughly. Okay, we're not going to talk about it now. The frontal lobe, that's your motor and personality. Okay, thinking, and intelligence, those kind of things, problem solving, and then motor. Because you actually use your motor system to solve problems in the world, right? So your thinking and motor system are very connected. And there's feelings that we grew this big frontal lobe because first of all, you have to explore your world to interact with your world. So your motor system, as you start to explore our world, we develop processing to make sense of that world. So anyway, so your motor and personality, intelligence, decision making, all there. Temporal lobe down here. Memory and auditory. Those are the two main functions. Just really good to just know what is big functions of these things, before you get it to the nitty-gritty. So auditory, it makes sense. The ears are kind of right there. It goes right in there. There's a primary auditory area like right there on the top of the temporal lobe where that initially auditory information goes into. And then deep inside there is the hippocampus, which is mainly memory for like factual knowledge, events, factual knowledge. Not motor memory. Motor memory is in your cerebellum and distributed, but and then parietal area, this is mainly for sensation. And then also a lot of association areas are in there. So you'll notice that the parietal lobe is strategically located. It has sides touching all the other lobes. It's the only lobe that has direct, connections or, approximation with all the other lobes. So visual information comes in, somatosensory information comes in, motor information that as you do movements, you get a little copy sent to your somatosensory area. Auditory information comes in, it's processed and integrated. So this is where a lot of integration of sensory and environmental information comes into your parietal lobe. Okay, so it's very integrative. And then from there, the parietal lobe will then often send signals to your frontal lobe to make decisions about, now what do we do with that information? How do we act? How do we change? What do we think about this? Occipital lobe, again, pretty big, and it's pretty much all vision back there. Vision, we're very visual creatures. We rely on vision very heavily, and so a lot of vision back there. And then remember I said there's that fifth lobe that people refer to called the insular lobe, or the limbic lobe. It's actually a region right in here. You actually have to this is that we call a lateral fissure, the lateral sylvian fissure right here. It's real big. If you take your tongs or your fingers and you pry it apart, you're going to see some more wrinkled gyri and sulci down in there. And that's sometimes people will call that the insular lobe. It's hidden until you pull it apart there. That's autonomic function and visceral function is down in there, is what it's, All right, perfect time for a little break before we name all these structures on the picture of a real brain. So let's come back, and again in about 10 minutes or so, 5 after. So brainstem. So we've been focusing on the cerebral hemispheres just most recently here. Now we're moving down to the brainstem. Has 3 main parts. Has a midbrain, has pons, and has a medulla. Sometimes the formal term is medulla oblongata. There's a movie you should watch. Well, you guys should have watched it last semester. Have you seen Gross Anatomy yet? Have you seen the movie? It's like 1980-something movie. You need to see it. It's like medical students going through anatomy, and there's just love drama and stress and whatever. And you guys would like totally identify with it, though. You guys, it's probably got to get it on VHS. I mean, I don't know if it's out. I'm sure you can probably get it on Netflix or something now. Gross Anatomy, but there's a anatomy professor who's guy that has an accent, and he's like medulla oblongata. He's like got some kind of a Jamaican accent or something. So you got to see it. What? Waterboy? Did he say that? Does they say that at Waterboy? Oh wow, there's another good reference I got to. Oh, there you go. Good. I need to add that clip into my, like Dr. Phil would have that clip up there. Like there. I got to get good at this. All right. So here, we've shown you. Here's the view, sagittal view, midbrain, pons, medulla. Now here's looking at a ventral or anterior view of the brain stem with everything else cut away. Okay, so cerebrum would be up here, the cerebral hemispheres would be up here. So this right here, that's your medulla. Sorry, midbrain. Midbrain is right there. This is your pons. This is we're looking at the ventral surface or the anterior surface of the pons here, which is often referred to as the basilar pons, but don't even know that yet. And then this almost somewhat triangular shaped structure here, that's your medulla down below that. Okay? And these, it's pretty easy to see the divisions here. You see that nice little line there in this bulbous structure, and you see a nice little line there. So it really is kind of easy to kind of know where one of these structures starts and when the other begins. Okay. So let's talk about the ventral surface and go into a little more detail on the surface anatomy on the front part of your brainstem. We do have cranial nerves. You're going to see 2 through 12 all exit from that. There is one little crazy cranial nerve number 4, your trochlear nerve. It's the only one that comes off the back of your brainstem. And it also crosses as it comes out, which is the little exception to the rule. Actually, you can see this little tag end right here? This little thing sticking out right there on the edge? That's the little tag end of your trochlear nerve probably coming off the back of your brainstem there. It's sticking out there on the side. So don't worry about that right now, but just notice that. So we'll show you in the next slide where those cranial nerves approximately exit or come off of, but let's focus on the front. So the first structure is in the midbrain. So here's our midbrain. This whole structure together is the midbrain. Okay. The most prominent ventral front feature of that is these guys right here. These structures right here on both sides. Those are the cerebral peduncles. Remember we said a peduncle? It's a connecting stalk. In that connecting stalk is a bunch of axons. So you actually have a bunch of axons that run through these cerebral peduncles and also go down through the front of your pons and the front of your medulla. Those are mostly motor axons. There's a bunch of motor axons that are traveling down through that front part of your brainstem, and they travel through the cerebral peduncles as they're on their way down to the pons and to the medulla, and then they cross over in the medulla and then go to the opposite side of your body. Okay. Remember I said there's more than one term for most everything. So in your reading you may see if they call it the crus cerebri is another term for the cerebral peduncles, or they call it the basis pedunculi. The peduncles at the base of your brain, the basis pedunculi, okay, is another term. Within there is a depression, a fossa, called the interpeduncular fossa. Okay. The only thing relevant about that is this is where your oculomotor nerve exits out of the interpeduncular fossa. Don't have to know that now, but that's where cranial nerve number 3 comes out of the interpeduncular fossa. And just in case you're wondering, these 2 bumps right here are called the mammillary bodies. And if you just let your imagination wander, you might see why they call them the mammillary bodies. We don't care about it as physical therapists. They don't even sure exactly what they do. They probably have some autonomic role or limbic function role, but so don't worry about the mammillary bodies, but they're just kind of hanging there in the way. They're kind of, they're distracting, I know. So, all right, interpeduncular fossa, we got that. Basilar pons and groove. So that's this front part of the pons, this ventral surface is referred to as the basilar pons, okay? And there's a subtle groove or depression right here that I'm kind of highlighting there. It's kind of hard to see from this two-dimensional image, but there's a little bit of a depression there. It's called the basilar groove because the basilar artery lays inside of that little groove there. There's a basilar artery laying right there. When we get to the blood supply, you'll see the basilar artery on there. Then we move down to the medulla, okay? The medulla, we've got two bumps right here called the olives. There's cell bodies in those olives, okay? They have direct connection with your cerebellum. They play a role in regulating coordination and movement, the olives. When we get to the cerebellum connections, you'll see where they send fibers into the cerebellum called the olivary fibers. They're almost like a mini cerebellum. They do some processing in them a little bit. The other main things you need to look at on the front part here is we have the medullary pyramids. You got one on each side here. You got your olives lateral. So olives are lateral to the medullary pyramids are more medial. Remember we talked about the motor fibers that run down the front part through your, coming down from your cerebrum. They go down through your cerebral peduncles, down through the basilar pons. They go into the pyramids. The front part of your medulla, the pyramids, is where the motor fibers are running. Then they eventually get down to this little point right here where they cross over. So these cross over, then go down into your spinal cord on the opposite side. And that's called the pyramidal decussation, okay? Or sometimes called the decussation of the pyramids, the pyramidal decussation. And if you, maybe I'll take a brainstem out this year and have a decent brainstem specimen for you to look at. But if you take like a probe, and you take your little probe and start running it down this little ventromedian fissure here in between the pyramids, you start running it down, and then all of a sudden you'll get down to this, like you can see it right there is where it starts. You'll start running down, boom, you hit a little stopping point. That's where those axons start crossing to go over to the other side. Those motor axons cross over, so it's kind of fun. Boom, and there it is. And that's where you run into the decussation of the pyramids at that point. All I want you to appreciate here is all the yellow things are cranial nerves. Don't have to know the numbers of them, don't have to know them yet, we'll get there. Just that they're all coming out of that, mainly that ventral and lateral parts of the brain stem, okay? Here's that oculomotor nerve right here. That's that interpeduncular fossa. This is the cerebral peduncles right here on each side. There's that interpeduncular fossa with that oculomotor nerve coming out of there. And that's just kind of interesting too. When you look at this on the real specimens tomorrow, one of the things we'll identify is this is your temporal lobe here. There's the most medial protrusion of your temporal lobe is this thing called the uncus. There's actually a membrane here, some dura, and what happens is that when your brain swells, as your brain gets pushed and swells, this uncus comes over the edge of that membrane and pushes on your oculomotor nerve. And when you block the oculomotor nerve, your pupil blows out and dilates, okay? Because this supplies it blows out your pupil. So by compressing that nerve. So that's when you're doing a quick check on the field or something, you see a blown pupil, that means you have swelling bad enough that uncus got pushed over the tentorial membrane there and is pushing on that oculomotor nerve. That means emergency, evacuation, burr hole, suck out blood. But now we're on the dorsal surface of the brainstem. It's got a little more interesting stuff going on here. This is your midbrain, right? It's kind of right in here is midbrain, all this area here. This is actually a little bit of your thalamus. The thalamus is the two egg-shaped structures right here. This is a little bit of your third ventricle, which is between the two thalami, but the thalamus has been cut off. We're looking at the back of the brainstem, so this is the medulla. Main thing we're going to see here is the superior colliculi. Let me get rid of some of this extra crap. There's a bump here and a bump here. That's a colliculus, a superior colliculus. And then there's another bump here, another bump here, inferior colliculus, or the inferior colliculi as a group, the superior colliculi. Underneath those bumps are little groups of cell bodies, okay? There's a group of cell bodies in here that is associated with your reflexive movement of your eyes towards a stimulus. Like in your peripheral field of vision, you know how like it's really hard, even when, like if you just see something in your periphery, like you almost, unless you're really consciously acting, your eyes will divert to that image. That mechanism is mediated by the brainstem and via this superior colliculus. So when you get a little visual disturbance or see something like, I better check that out, make sure it's not something dangerous or threatening, you do a quick little look with your eye before it ever gets to your cortex. You don't even know what that image is. Your eyes are already going to it. By the time you get there, then you recognize what it is. But a signal is sent to that inferior colliculus. That inferior colliculus processes that really quick, little minor processing. There's cell bodies in there. Then it sends axons down to your neck muscles, directly down to your neck muscles to get your head turning there before the image has even gotten to your brain. Your head's already turning towards it. And that's mediated through this little superior colliculi, and there's actually a little pathway of fibers called the medial longitudinal fasciculus that we'll talk about, the MLF, which connects your superior inferior colliculi with your upper neck muscles to do responses to both visual information that you want to like take a quick look at, reflexive vision kind of responses, and also auditory. You have the same mechanism like you hear a loud sound. You have to, you kind of have to turn to look at it. That's mediated by the inferior colliculus down here. Same kind of function is that reflexive movement of your head and neck towards an auditory stimulus is through the inferior colliculus. And I kind of think my eyes are a little higher than my ear canals, so that's how I remember superior colliculus is my eyes and my inferior colliculus is my ears. So that's how I remember it anyway. My tricks may not work for you. Mine may be really dumb and silly, but sometimes they work. Remember I said there's that one cranial nerve that comes off the back of the, here it is, and that's that little tag end I was showing you from the back end. This is the trochlear nerve. And it goes to innervate one little muscle called the superior oblique muscle in your eye, which makes your eye go down and in a little bit. So that's the superior oblique, that cranial nerve number 4. And it's really crazy that the cell body for this one is actually located over here. The cell body for this one's deep inside there, and it crosses over. That's the only cranial nerve that crosses. All the rest of your cranial nerves, the nuclei, this is the trochlear nucleus right here. A nucleus is a group of cell bodies in your central nervous system. Your brainstem is central nervous system. So the trochlear nucleus is over here, and it projects its axon and crosses over when it comes out, where all the other cranial nerve nuclei, like our trigeminal nerve down here, it comes out, the nucleus and the axons come out on the same side. Don't have to know that now, but just kind of fun fact. It is kind of interesting, isn't it? I mean, I'm trying to make it a little bit interesting, neuro. Believe me, I have to do something so I don't bore myself to tears either. So, rhomboid fossa is the next thing down here. So this is your pons right in here. Okay, that's the pons. there's a structure that actually is both in the pons and the medulla. It's called the rhomboid fossa. So it's a depression that is somewhat rhomboid looking in nature, you know. Looks like a rhomboid. And actually this particular specimen, there's this little structure right up here. You can kind of see it. This little thing right here is this little covering, or velum it's called, that's covering over. If you could cut that out, then it'd even look more rhomboid looking in nature if you cut that little velum out of there, to make it, because it's still hollow up underneath there. But the rhomboid fossa, you can't see it unless you got to cut the cerebellum off the back. You got to cut the cerebellum sitting back behind the pons and medulla right there. So the cerebellum has two hemispheres, and it's sitting off the back here. It's been cut off and pulled away, and we're looking at the back side, that posterior or dorsal surface. This would be cut. We call this a posterior or dorsal surface because the brainstem's in line with the body's axis here. The neuraxis is in line with the body's axis here. Okay. When you cut off the cerebellum, it is connected to the brainstem via peduncles. Kind of like your cerebrum is connected to the midbrain via peduncles, your cerebellum is stuck on the back of the brainstem via peduncles. And depending on how you cut it, you can actually identify three separate peduncles. There is a superior cerebellar peduncle, a middle cerebellar peduncle, and an inferior cerebellar peduncle. It's inferior because it's just a little bit, it's the one that goes most inferior. So on this side, this would be the superior, inferior, and the middle cerebellar peduncles. In order for you guys to appreciate those, if I cut a cerebellum off the back, I have to do those angles slightly differently so you can appreciate. If I just did a flat cut, it'd be hard to recognize where those are, but there is three separate peduncles sitting off the back. Don't have to know all those right this moment, but just pointing things out, so you hear it 5 times, you know, eventually. And it'll stick on the 5th, hopefully, before the exam. So those are the peduncles, okay? Because what you have is, so when you when you are sending a motor signal down to go do something, so remember I said it goes down the front of your brainstem, you know, crosses over at that desiccation, all that. But you also send a copy to your cerebellum. And so the copy goes, it's coming down from your cerebrum, it goes through the superior cerebellar peduncle into your cerebellum there. And then when you actually start doing some activity and your muscle moves your joint and your limbs, you have sensory information that says, well, how much did my joint move? It comes back and goes, the sensory information comes back, and a copy of that information is delivered to your cerebellum via the inferior cerebellar peduncle. Your body compares that information. I'm sorry, I messed up. Initially, that initial signal comes down and goes through your middle cerebellar peduncle to go to your cerebellum. The sensory information coming back that says what actually happened when you moved your limb, you know, because maybe it was a heavier thing you thought. Maybe you didn't generate enough force to lift that object, or it moved faster than you expected, and your proprioceptors are telling you, oh, you need to generate more force. Well, you send the proprioceptive signal up here. These guys compare each other. They say, hey, did the signal I originally sent, did it match with what I expected to happen? And if it says, ooh, there's a mismatch here, it sends a signal through the superior cerebellar peduncle back up to the cerebrum to then adjust the next signal you send back down to, like, let's ramp that up the next time. You know, let's ramp it down a little bit. That was too much force, not enough force. So don't have to know that right now. Just showing you why you have those 3 peduncles, and it's really very much like just electrical. It's like just signal flow through these areas, and some of that anatomy, it makes sense why it would go through those different areas. How it gets in, how axons get in and out of the cerebellum is through those peduncles. That's all you got to know right now. On the back of your brainstem, so here's medulla, there are some small tubercles. Remember, tubercles are those small bumps. There's a little tubercle right here, a little tubercle right there. There's another one here, another one there. There are pairs of them, okay? You have The gracile tubercle is the more medial one, gracile, and the more lateral is the cuneate tubercle. Underneath that tubercle is, if we do a cross section and look inside, so the tissue inside of there, deep inside of there, that's where the gracile nucleus and the cuneate nuclei are lying. The tubercle is just the surface feature, but the actual, nuclei and the cell bodies are deep in there. Okay, but they create this bump on the outside because there's a group of them that protrude it out a little bit. And those are a relay. Those are one of the connections in your somatosensory pathways for proprioception. The cuneate tubercle has to do with proprioception coming from your upper body. The gracile tubercle, you have a gracilis muscle. That's how I remember it's coming. That has to do with lower body proprioceptive information. We're going to get all into every one of those connections when we get to the sensory systems and go through those pathways. Don't have to know it now. Gracile and cuneate tubercles. There's also, I don't have it listed here. So here's the tubercle. You can see these little divisions right here. Here's a dorsal median fissure here. Then right here is an intermediate one. There's another intermediate one here, and there's just a little bit of division here you can see too. So there's actually columns back here called the dorsal columns that are running up. And there's axons in these dorsal columns that are running up to go connect to these nuclei that are sitting below those tubercles or back underneath those tubercles. That's all sensory information, and this actually all is proprioceptive information. So let me just point out one at a time here. So this little thing right here, that's called the fasciculus cuneatus. Remember, fasciculus is just a group of axons. So there's a group of axons there that's running up the back of your brainstem, and that's bringing proprioceptive information from your upper body that it's going to go and it's going to talk to the nucleus cuneatus right there. Okay, and then the dorsal column that's more medial is this guy right here. That's your fasciculus gracilis. And that's where axons that are carrying proprioceptive information from your lower body are going to go up and connect with the nucleus gracilis, which is just below the gracile tubercle. Make sense? But right now, this is all you have to know about that. Cerebellum, that structure sitting off the back of the brainstem. Incredible structure for us as therapists because it's the thing that primary function is coordination of movement, and we'll get into a little bit. It's just phenomenal that what this thing does. It's like the like a car now has your computer module, like your adaptive cruise control, your anti-lock brakes, all that stuff is like your cerebellum of your body. Your body it does all that without you thinking about it. It's all happening in the background. It's making all these adjustments. And so the cerebellum is like that for your body. And all I want, appreciate the structure. Look how folded it is. How many sulci and gyri. Anybody know what the purpose of why your brain folds back in on itself, or why your cerebellum would do that? It's kind of like. So yeah, if you just have a flat structure, there's only so much surface area. If you start folding yourself back on each other, it increases the amount of surface area that you can actually have more neurons and more processing power. And so when you look at the cerebellum, it's even more folded up and has more ridges and sulci and everything than your actual cerebrum. So it has massive processing power to control your body in space and help you coordinate every movement of your body on the fly, and it's just phenomenal. So in the sagittal view like this, we can identify an anterior lobe and a posterior lobe. There's a pretty big division right here. So this is the anterior lobe, and all the rest of here is the posterior lobe. So about 1/3 of it's anterior lobe, 2/3 your cerebrum or your cerebellum is posterior lobe. A little harder to see this split on this is a superior, okay, look down on top of the cerebellum. So the cerebrum has been taken, you know, we're looking straight down on top of the cerebellum. The cerebrum has been taken away, and we've taken the brainstem off. That division is right there. This is the anterior lobe, then all the rest of this back here is the posterior lobe. So we're looking down this way, you know. This is anterior, posterior that we're looking at. That's the division, anterior and posterior we're looking at there, okay? From the front, now this down here in that left-hand corner there is looking at the ventral surface or anterior surface of this cerebellum as it's been cut off of the back of the brainstem. And we can identify those peduncles again, because remember those peduncles that have been cut through to take it off of there? So here's the superior cerebellar peduncle. This is the inferior cerebellar peduncle, and this is the middle cerebellar peduncle. Superior, inferior, and middle cerebellar peduncles. Okay? And when your cerebellum is actually attached to your brainstem, it actually forms a little space. And that space is the fourth ventricle. There's cerebrospinal fluid in there. And so your cerebellum is the roof of your fourth ventricle. And that rhomboid fossa, when you pull that off, that's the floor of your fourth ventricle. They call that the floor of your fourth ventricle. So you pop that off, and your cerebellum's the roof, and the rhomboid fossa is the floor of your fourth ventricle. You have to have the two together to have the space to have the fourth ventricle. It's kind of like the two cerebral hemispheres. They have to come together. There's a little tiny sliver of space between the two cerebral hemispheres, between the thalami on each side. That's the third ventricle. It's right in between there. The fourth ventricle sits down below that. And we'll get to the ventricular system here in a minute. Real briefly, we're going to go through spinal cord. We're going to get all this again. You guys know the spinal cord a little bit because of peripheral and the spinal cord, spinal nerve roots. Lies within the vertebral canal, extends to the level of the L1, L2 intervertebral disc. Very important to know the spinal cord doesn't go all the way down. It ends at L1, L2, okay? It ends as and then you have the cauda equina, which is the rest of the nerve roots that are going down to all your lower lumbar and sacral nerve roots, because they all have to come off from up that L1, L2 region. They got to go, but they got to extend all the way down to come out at their foramina at the appropriate level. So that's the cauda equina. It looks like a horse's tail when you kind of just dangle it and spread it out kind of thing. There's also a little tiny structure. There's a little thing called the filum terminale, which is this little extension of pia mater, which is one of your glial cells that's a covering of your nervous system, and it extends down and anchors your spinal cord to your sacrum. There's this thing called the filum terminale. It's so delicate and fine, it just seems like it would just snap or break, you know. It's kind of interesting. We have dorsal and ventral roots we talked about that come off your spinal cord to form your spinal nerves. You know, the ventral root is associated with motor function. That's where your axons that go to your motor come out, and the ventral roots is where you bring in sensory information comes into the spinal cord towards the backside via the dorsal roots. 31 pairs spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, 1 coccygeal. Here's a sagittal view to start with, and all I want you to see here is here's that termination right here between the L1, L2 disc space is where the tip of your spinal cord ends. And then those nerve roots that all come off that end there have to extend down to exit their appropriate spinal levels. Okay. There's some clinical importance here when we talk about injuries to the spinal column, spinal cord, is that if you break your back down here below the level where that spinal cord ends, that gives you a whole different type of symptoms than if you break your back or neck where you actually damage the spinal cord. Okay, one's upper motor neuron types of deficits, one's lower motor neuron types of deficits. One's called cauda equina syndrome is the syndrome, but we don't need to know that now. But just kind of. It's just from a clinical standpoint, you see all the extra. There's a lot of space actually in here. Once you get rid of the spinal cord, this kind of opens up. But that's why you can sort of safely, put needles in there and like take people's cerebrospinal fluid or give them an epidural, because most of the time those nerves get out of the way as you poke the needle in there. Occasionally you'll nick one, and then somebody ends up with some significant pain and some complications. That gets better that once the myelin kind of regenerates and so forth. But that's how you can get into lumbar punctures and those kind of things. So. There's the cauda. So this whole area is the cauda equina, the horse's tail, and then there's a little filum terminale that extends down and anchors, to your sacrum down here. That little extension of pia mater, which is the most closest covering of your nervous system. You have 3 layers of covering: pia, arachnoid, and dura. The pia extends off there and anchors it. This is actually looking in a cadaver section. You've done a laminectomy throughout the, laminotomy for the whole thing, and just pulled off all the posterior part of the vertebral column there. And all these are those. This is the tip of your spinal cord right here. And this is all the cauda equina, and then you can see filum terminale is that little lighter structure right there, and it's going to come down. This is your sacrum right here. It's going to anchor that right to your sacrum there. Spinal cord, internal features. When you take a cross section through a spinal cord, so a horizontal section through a spinal cord, you have gray matter and white matter that you'll see. The gray matter in the spinal cord is on the inside. It's the opposite of the cerebrum. Your cerebrum, most of your gray matter is on the outside, except for the basal ganglia and the thalamus. But in the spinal cord, all your gray matter, all your cell bodies are in the inside. All the white matter, the axons that are running up and down, are on the outside. Okay? That gray matter, we can talk about dorsal horns. Let me go to a little darker color. So dorsal horns, so this is the back, this is dorsum or dorsal area, and this is front, anterior ventral, because we're in the same in line with the body's axis. Dorsal horns, anterior horns, and then there are what's called the lateral horns, but the lateral horns are only in the thoracolumbar region because this is where your sympathetic nerves come off of, okay? Don't need to know that necessarily now, but if you see a lateral horn, if you see a cross-section of a spinal cord and you see a lateral horn, you know you've got to be somewhere between, T1 and L1, you got to be somewhere in that region because that's where your thoracolumbar, that's where sympathetic nerves come out, and that's what forms your lateral horns, is that's where the cell bodies for the autonomic sympathetic nerves are sitting. Then the axons come out of there. So this would be obviously a thoracolumbar segment somewhere. Okay. And the white matter, that's all the rest on the outside here. Remember we talked about fasciculi, where the little bundles or packets, the funiculi. So the spinal cord we can actually divide into these bigger, like this whole big part of white matter back here, that's the posterior funiculus. This big area right out here, that's the lateral funiculus, but it's going to be divided into smaller regions and pathways that are located. And then this area right here, this is the anterior funiculus. Don't have to know that now. Just here's your spinal cord actually within the vertebral column in a horizontal section here. Here is the spinal cord. Here's your dorsal horn. This is the dorsal root, okay? There's a little bump here, a little larger structure. That's the dorsal root ganglia. So this is bringing in sensory information, comes into the back part of the spinal cord. The cell body for this long axon that goes and creates that nerve is located outside of the central nervous system as the dorsal root ganglia, okay, where the cell body is, okay? The ventral root, here's the ventral root. These are motor axons that come out and go out the ventral root. Then eventually they all come together as the spinal nerve, right? Then they're all packaged together and going to wherever they need to go. This happens to be in the thoracolumbar area, so because we see a little tiny lateral horn there, so there is some autonomic function. The autonomic fibers also go out the ventral root. Then they go into this little area here. There's actually a ganglia down here. Paravertebral ganglia, it makes a little connection here, then it goes back out and joins up with the motor and sensory information all coming through that nerve, okay? Once you get past that, once you get out of the foramina, there's the hole, the foramina, everything's together. These are some of the coverings here. This is the pia, that little purple layer, right? That's a microscopic layer that lines all of your nervous system, spinal cord, brain, brainstem. The next layer out is the arachnoid. The next layer out is the dura. Don't need to know that at the moment. which we're going to talk about now, the coverings of the nervous system. This is like the big, big overview today. It's a lot of information. This will seem really easy when we get later on, but it's going to take a little time. It's just like I'm hitting you with a lot, just kind of like most of the stuff you may have heard once or twice before, but just, all right, let's kind of reorient yourself. So, coverings of the nervous system, collectively known as the meninges. We refer to these as the meninges. And these are made up of those support cells, glial type cells, okay, as the meninges. 3 layers. These surround your entire central nervous system, not your peripheral nervous system. So this is just brain, brainstem, spinal cord. Your entire brain, brainstem, and spinal cord is surrounded by these 3 layers. The outermost layer is the dura. It's very tough. It's kind of shiny. It's kind of fibrous, and you'll see it when I bring in a specimen or we take a brain out. It's really hard. It's even hard to cut through sometimes. It's almost like a tendon, almost. It's really like a fascia, like a really thick fascia. So what it's more like is like a fascia. It's the dura. It's the outermost membrane. It actually forms like the falx cerebri. So your dura goes down between your 2 cerebral hemispheres and forms this thing called the falx cerebri. It forms the tentorium. There's like a little layer of dura that goes between your bottom of your cerebrum and the top of your cerebellum. And that's what that little uncus gets pushed over when you blow out and you pushes on your nerve is this tentorium. It's like a tent over your cerebellum, the tentorium cerebelli. It makes sense. Tentorium, it's a tent over your cerebellum. It's where the dura goes in between your cerebrum and your cerebellum and forms a little tent over it. It also forms the dural sinuses, okay, the venous sinuses. So eventually your venous blood within your brain, all of it eventually gets dumped into the dural sinuses, or like big drainage ditches, basically. The dural sinuses then take it out. They eventually go into your jugular vein to go recycled back in. So the venous blood goes from your little veins and venules in your cerebrum, then eventually goes into these dural sinuses to make it back in your jugular vein. So that's the dura makes up these. The arachnoid is the next layer deep. Okay, the arachnoid layer, arachnoid, spider-like, and when you take the dura off, you see this thin, fibrous-looking, see-through, almost can look like a spider-webby thing over everything else. And there will be, on all the brains tomorrow, there'll be some arachnoid left somewhere on those. It's hard to get off. It's really kind of really close to the, and it's really slippery, and it's hard to get off with gloves. I got to take like a little, pick and kind of like stick it in and kind of pull it away, but it tears. And usually what you have to do is kind of grab all the blood vessels that are on the surface of the cerebrum and pull that off. And usually as you pull all the blood vessels off, the arachnoid comes with it. Because there's a little space called the subarachnoid space, and you have to have cerebral spinal fluid in your system to have a subarachnoid space. Cerebral spinal fluid fills up that space and keeps it puffed up, and so that the arachnoid and the pia, which is the deepest layer, which is right on top of the nervous system, there's a little space there, and when you're producing cerebral spinal fluid, it keeps that full. I mean, we're talking a millimeter or two of space there. And that insulates and cushions your nervous system a little bit in that subarachnoid space. Within that subarachnoid space is where a lot of your surface arteries are. So if you ever heard the term have a subarachnoid bleed, that means one of the arteries that is on the surface of your brain or brainstem is bleeding into that subarachnoid space. That's a bad bleed. You don't want a subarachnoid hemorrhage, because that creates pressure really quick. Those are pretty high pressure arteries still. They're pretty big still on the surface of your cerebrum, and that's a subarachnoid bleed. Okay? That's also where your cerebral spinal fluid is flowing in that subarachnoid space. So your whole central nervous system is floating in that subarachnoid, in that cerebral spinal fluid that's contained in that subarachnoid space, and it's flowing through there and being recycled. And we'll show you how that process works. Then that last, most inner layer is the pia. The pia mater is the delicate membrane of microscopic thickness. You can't see the pia. You need a microscope to see the pia. Unless you're looking at the filum terminale. Then that's actually pia that's now big enough you can see to form that filum terminale. The pia also forms what's called the dentate ligaments, which off your spinal cord at each level, there's these little extensions of pia that come off to anchor your spinal cord at the sides too. So you have the filum terminale that anchors it at the sacrum, and then you have these little called dentate ligaments, which are pia that come off the side of your spinal cord to anchor it to the vertebral levels side to side. So this is looking through like a cross section, like a horizontal, a coronal section through your cerebrum here. And you're looking at, here's the white matter down here, the deep white matter. Here's your cortex, which is all your cell bodies for all your neurons. And then here is this most surface layer here, the closest one, that microscope, that's the pia. And then the next layer is the arachnoid, and it has these little end feet too, but there's a little tiny space. See that little space in there? That's where the cerebral spinal fluid is running, and that's where those surface arteries are also sitting. See those surface arteries that are sitting there in the sulci? They run in the sulci of your cerebrum. So almost every sulci you have, there's a little artery running in that sulci. And so sometimes I can get in there and start pulling those arteries out of the sulci, and usually when I do that, the arachnoid comes with it. And then lastly, here's the dura, the really thick, and that's adhered to the bone. That dura is typically adhered to the bone. So what we've done here is, normally this whole brain would be covered with this glistening dura mater. All right. And what we've done is we've cut some of that dura mater off to expose where the dura penetrates into these big gaps between your cerebral longitudinal fissure and between where your cerebellum and your bottom of your cerebrum meet. So it dives down in here, and so this is the falx cerebri. So there's this, and it's really kind of cool when it attaches to the front of your cranium and the crista galli, and if you cut that, I can pull that whole thing out, and you see this little, C-shaped looking falx cerebri that sits right down in the middle between and it's just dura that dives down in between. And then here's that tentorium cerebelli. It's this structure right here, and it kind of comes up like this. So it's got a little bit of a tent-like structure, and it's separating between the top of your cerebellum and the bottom of your cerebrum here. It's just a little separating there. Dives in there. This is that falx. Here's the falx cerebri right here. It's this glistening, fibrous-looking structure here. Then here's the tentorium cerebelli. See, it comes up all the way up in between here, between the bottom of your cerebrum and the top of your cerebellum. And it actually kind of extends out this way, and then this is where your cranial nerve 3 comes out of that area, and that's where that uncus can push on it there. So the tentorium cerebelli extends all the way like this. You wouldn't see it there because obviously it's on the outside. It's not cutting through your nervous system. I should draw that in dotted lines there. So it's an external structure. Blood supply. A couple quick things on that. We'll be good to go. Long day, I know, first day. You got a couple main supplies of blood to your cerebrum, or to your central nervous system, brainstem and cerebrum. The internal carotids are the big ones for the cerebrum. They provide most of the blood to the cerebral hemispheres. They basically divide into a middle and anterior cerebral artery, as there are two big branches they first divide into. They divide into lots of sub-branches, but all we need to know right now is internal carotids divide into those two main middle and anterior cerebral arteries. The vertebral arteries, which you guys have probably already talked about, right? A little bit, vertebral arteries, they're running up along your vertebrae, and those are the ones, if you snap somebody's neck, you could compromise or torque or twist. Those are going to the occipital region of your cerebrum, mainly the lower occipital, the underneath surface of your cerebrum. So most of your cerebrum is internal carotids. But the bottom part of your cerebrum and the occipital lobe has vertebral artery circulation goes to those, as well as the brainstem region. The vertebral arteries come up and they join to form the basilar artery. Remember, that lies in that basilar groove in the front of the brainstem. The dural sinuses, I mentioned, drain venous blood from the brain, eventually the dural sinuses, which is kind of a unique feature. Here's your internal carotids that I just circled right there. So internal carotids, those are coming up through the front part of your neck, go up up in this region towards the front part of your brain, just here in front of the temporal lobes there. They divide into your middle and anterior cerebral arteries to supply most of the top half and outer surface of your cerebrum. Here's the vertebral arteries coming up. They come up the brainstem, they join to form this big basilar artery, then they actually terminate as the posterior cerebral arteries. But they also have some branches that they supply the cerebellum, the brainstem, and also some of this underneath surface of the cerebrum and back part of the cerebrum. That's just where they're closest to, and you just can see that. Don't worry about this yet. That's what it looks like in real life when it's actually been colorized. That's been colorized. They've colorized the vasculature there to make it even easier to see. It used to be I would find those things in a real specimen and tag those. You'd have a pin going through one of those. I don't do that to you guys. I don't do it to you guys anymore. That was the good old days. I think we're almost done here. Ventricular, are we getting the last stuff, aren't we? Ventricular system, or sort of, well, a little bit more. We're going to finish after ventricular system here. So the ventricles are these hollow areas within and deep inside your cerebrum and your brainstem that contains cerebrospinal fluid. This is where your cerebrospinal fluid starts, where it's generated. That cerebrospinal fluid eventually gets out of the ventricles to go into your subarachnoid space. We'll talk about how it gets out of here. It starts inside your brain, but it has to get outside to go to the outside to circulate around the outside of your cerebrum. So the primary function of the ventricular system is the regulation of extracellular fluid and some protection of the CNS, because the ventricles are going to produce that cerebrospinal fluid that are going to help protect your CNS. The ability to have those spaces also allows your brain to swell or contract a little bit. If your brain starts to swell, your ventricles can shrink a little bit to accommodate that. You can just produce less cerebrospinal fluid temporarily. And if your brain starts to atrophy, your ventricles just get bigger and you just make more cerebrospinal fluid to take up that space. So when we look at a brain that has a lot of like Alzheimer's or dementia, and there's a lot of atrophy of their brain, they have these massive ventricles. Because they just, you lose nervous system tissue, your ventricles just expand into that and you just have more cerebrospinal fluid in there. So. The choroid plexus is this structure that forms the cerebrospinal fluid. It's like it's very granular in nature. It's like these little capillaries that strain the blood to make, basically that's like a plasma. Your CSF is very much like plasma, and that's how you generate this, is you just filter your blood via the choroid plexus to generate this CSF. And most of the brains will have a little bit of choroid plexus in there I can point out for you guys. You have two lateral ventricles. That's these big C-shaped guys. You have one in each hemisphere. They're the other, so you have a left and right that I just circled there. So you have a pair of lateral ventricles. That's your 1 and 2. They're just two lateral ventricles. Then you have a third ventricle, which is this space right in here. It's that little sliver of space between where the two thalamuses are, okay? There's a little thing right here you'll see. There's actually a little connecting stalk between your two thalami called the thalamic adhesion. And so that's why they show there's no space there, because there's a connection there. But there's space all around that. And you have a thalamus on each side with this little tiny stalk in there that's poking through the middle of your third ventricle. That's what that little hole is this connecting stalk between there. So this is your third ventricle here. And so cerebrospinal fluid goes from your lateral ventricles. There's a little connection right there. It's called the interventricular foramen, or the foramen of Monro. It goes from your lateral ventricle into your third ventricle. Okay? And third ventricle gets it from both lateral ventricles. There's a little connecting little aqueduct there, not aqueduct, but the foramen that connects those. Both go dump into that third ventricle. So both lateral ventricles dump into the third ventricle. And then that cerebrospinal fluid goes down through this thing called the cerebral aqueduct, which is in your midbrain. It's a little tiny tube that goes down and eventually gets back into here. This is your fourth ventricle. Remember, the fourth ventricle is formed by the cerebellum and the brainstem here. Between those, it's kind of between the two, the cerebellum and the brainstem. That space between them is the fourth ventricle. And then eventually, there are 3 openings at the base of your cerebellum where the cerebrospinal fluid squirts out and gets into your subarachnoid space. There's a little opening here on each side. Those are called the foramina luschka. Don't need to know that right now. Okay. Or the lateral apertures, the foramina luschka. There's two little holes on the outside lateral surfaces just underneath the bottom of your cerebellum. That cerebrospinal fluid gets out into that subarachnoid space. And there's I don't have a view of it. You have to look right from the back of the cerebellum, just underneath the very bottom of your cerebellum on the back is another little hole underneath the bottom there called the foramina magendi. Okay. And that's where it leaks out there too, to get into your subarachnoid space. So if you block that cerebral aqueduct, or you block one of these connections, or you block those holes below the cerebellum, you get a backup in your system. And that's where you get what's called hydrocephalus, where the brain starts to expand. If you're a young child who doesn't have fused skull, your skull grows. If you're an adult, you have pressure that builds up in your head that eventually you have to have released, and you actually have to then put in a valve and a drain. So dural drain there to drain that. They just the tube goes down through your brain. They'd find a spot that doesn't look like it's going to cause. There's spots of your brain you can poke small tubes that's not going to cause you paralysis or thinking problems, and they poke that down through there, and then they run the tube through underneath your skin, down your neck, into your peritoneal cavity, so that just extra cerebrospinal fluid will just go down into your peritoneal, and your body reabsorbs it. And problem is those things get clogged occasionally, develop like little clots of protein builds up in those, and they have to replace them occasionally and those kind of things. So if you're working with kids, you'll have kids that have some of those, and you'll see the little knob or button up here that like where that junction is, where the tube starts, and you can actually palpate the tube going down in their neck, and then it goes underneath their clavicle. But you're just going to be careful when you're working with their little heads and necks that you don't. I mean, you can't hurt it really unless you just sit there and hold it for 3 hours or something like that, you know. But we're done. 1 minute late. I apologize. I owe you a minute for tomorrow. I'll get you out of promise a minute early tomorrow. Thanks for a good first day. Thanks for behaving. Thanks for not throwing anything at me. Put just as you leave, just stick your little information sheets at the end of the aisle. Yeah, just set them there. I'll come pick them up.